So the first note is that we're recording the webinar. So if you have questions and things to ask, just be mindful that that's being recorded. And also to say, I don't think we're going to be that numerous. So if you've got questions, feel free to put them in the Q&A or the chat during the webinar. Lenny Tatelman, the CEO, is in the background here today. So one of us, he may be able to answer while I'm talking, or we will get to those questions afterwards. So please feel free to do that as and when questions occur to you. So today will be very introductory to give you a glimpse of protocols.io and why it's important, we think, to share methods. It will not go hugely into functionality, but we'll give you links to sign up for demos, and or there will be other webinars that will give more insights into fuller, more detailed functionality and how to use the system. So please feel free to sign up for those at another time, but also email anytime if you have questions. I see people are still just joining, so we've just covered a little bit of housekeeping, but I'm about to kind of get started now, and the webinar is being recorded. So brief overview of what we'll cover, why it's important to share methods, a little bit about the mission and protocols.io and how we exist, and then a brief overview of functionality and the different ways that you would interact with protocols.io. We offer a protocol entry service, so I'll give you a pointer to that too, and then we should have time for questions. So if we start to think about why it's important to share methods, this is a really common problem for researchers, that you would be looking at a research paper, and in the materials and methods section, it may say something like, as described in X et al., another earlier paper that has been published. And if we go to the earlier paper, it may well be that you are pointed to another earlier paper. So this is a really common problem, and it's a source of great frustration. It also may be that if you contact those researchers, any full detail that should have been in a lab notebook from a previous postdoc might be lost. And this is a real problem, that researchers can't find, access and replicate experiments that have previously been published. So this tweet was from a biologist, but it's not just limited to biologists. There's another similar one here from a physicist who was looking for how devices were fabricated and ended up on exactly the same goose chase eventually to learn that devices were fabricated with conventional methods. So we've been using these tweets for a long time at protocols.io. So I've just been poking around, and there are a lot of more recent ones too that exist. So since those 2017, 2018 tweets, this problem has not miraculously disappeared. There are many people who are still incredibly frustrated at the fact that they can't get to the original methods. And why do we care? What is it important? There was an initiative by the Centre for Open Science a few years back, the Cancer Biology Reproducibility Project. They wanted to replicate the experiments from 50 cancer papers, high impact cancer papers. And there's a brilliant write-up by Ed Yong in The Atlantic, and I've kind of highlighted here in bold some of the key sections from this, where after talking to the people involved, the hardest part by far was figuring out exactly what the original labs actually did. So papers come with method sections that ought to provide recipes for doing the same experiments, but often these recipes are incomplete, missing out important steps, details, or ingredients. In some cases, the recipes aren't described at all. Long story short, they had to halt this effort after they'd attempted 18 of those 50 papers, replication of the experiments, because it was too time and money expensive to try to get the information. And they looked in the earlier papers, but they also talked to the researchers. So it turns out that, you know, even the researchers themselves do not have the information if they wanted to repeat the experiments in their own labs. So arguably that research dollars that have been spent supporting that is not best spent if we can't replicate down the line. So why does it matter? We're doing now quite a good job of capturing a lot of research data out there. So here I would propose that the cookies in this photo are the data. So these cookies are the result of tweaks to the very same recipe. So if we want to understand why these cookies are so phenotypically different, if we want to analyze the size and the thickness and the texture, we can actually only interpret those if we know what the tweaks were to the method. So we can only understand the differences in the data if we know how they were generated. So if we think about that in a bigger scale, all of the data that we're now carefully storing and capturing from research will only be useful in future if we have all of the information about how those data were generated. So I love this quote from an evolutionary biologist who said that nothing in biology makes sense except in the light of evolution, but I'm changing it now. So I would edit this one to nothing in research data makes sense except in the light of how it was generated. So if we're doing open research and open science and we're sharing our data, we need to make sure that we're sharing the comprehensive details of the methods as well. And this is where protocolsio comes in. Our mission is really simple. We want to make it easy to share method details before, during, and after publication. And so people may have heard the FAIR acronym: findable, accessible, interoperable, and reusable. Protocolsio is a repository that ticks pretty much all of those boxes. It's everything that's published on protocolsio has a digital object identifier, which is the same kind of identifier that any published research article receives. And they're all always open access, published with a CC BY license, which means they can always be reused by anyone. Author retains copyright, but anyone can reuse with attribution. It's a tool that can be used across all different research disciplines. So although originally created with kind of wet lab bench research in mind, these days it's incredibly versatile and is used by all different research disciplines, kind of from social sciences all the way across through the life sciences, biomedical, into clinical trials and public health, etc. We have a large number now of users, well over 100,000. Over 11,000 public protocols, but also very close to 40,000 now private protocols. So the private side is where researchers can collaborate to develop and optimize protocols before they make them public. And every protocol that's created on protocolsio starts out private, so you don't have to share until you're ready to do so. As a tool, it then really gives you a great ability to manage and share research data and protocols. It will simplify teamwork and improve collaboration. It works very much like any kind of Google Doc or spreadsheet where you can choose who to share with, collaborate, concurrently edit, so on. And if you use it throughout the research process, it really can save time and help you keep your work organized. There are a lot of organizations that already encourage use of protocolsio. So if you're a little bit kind of worried to understand whether journals will mind that you put the methods on a tool like protocolsio, actually, if you look at their author instructions, most of them are actively recommending that you use tools like protocolsio to share the methods, which means that the research they're publishing will be more reproducible. We have a lot of funders too who have either strong encouragements in their author kind of guidelines, their fundee guidelines, and some are much more strict and really require sharing of the methods on protocolsio. And then lastly here, we offer site licenses to institutions. So anyone can use protocolsio for free, but you would have a limited number of 5 private protocols, but you can publish. So when you publish one, you would be allowed another private one, and so on. Institutions can purchase a site license, which means that researchers in those locations can have unlimited number of protocols and workspaces. So as I mentioned, a lot of journals are actively encouraging you to use protocols.io in the context of the research article, and this is how that might look. So here's an example from a PLOS biology paper. You can see in the materials and methods section, they note that methods and protocols for Drosophila rearing, media preps, and microbial manipulations are available as a collection in protocols.io. And then they have the DOI, that permanent link to the collection on protocols.io. So if you click through to the link, you come to protocols.io, and you will see the collection in the background. And if the authors may have updated and added a new version to their protocol collection, you will automatically be alerted if there is a new version. So we also think of this as kind of dynamic permanence, and that the researcher has a lot more control, because if you realize you can better clarify how to perform the experiments, or there was a slight error in one of the protocols that you put live with the paper, the researcher can really easily create their own new version of the protocol, and anyone who comes to view will always be told if there's a newer version. So you would click to see the new version, and then there's the protocol collection. You can always compare back to earlier versions, so you'll be able to see what's changed if you're wondering what happened from version 1 to version 2. We also have a much more active partnership with PLOS ONE, who have now a new article type, well, new, I guess it's a year old, almost to the day. We launched this last February 2021, a new article type, lab protocols. And these are very much to publish the methods themselves slightly separately from the data research article. And you can see that from the research article, there's a very prominent link to see the protocol. And then similarly on the protocol, it links back to the PLOS ONE article. So the materials and methods section in these articles is a protocol on protocols.io, a protocol or a collection of protocols. And so this means that sorry, jumping around the slides, that the person who created the method can receive credit for the method itself. So it may be that you spend your PhD or postdoc honing and optimizing a method, but you never get around to generating a lot of research results with that method. This way you can get the appropriate credit for the method, which can be, you know, the most arduous part of the process, as a distinct output, research output. So I'll go on now to talk a little bit more about protocols.io itself. So I mentioned this already, that it's an open access repository and it's free to publish and get a DOI, and anyone globally, anywhere can come, create an account and start making protocols and publishing protocols. But then we do offer the premium workspaces. So that's the paid offering that can happen at institution level or just at a group level if you wanted to share with a small number of colleagues and have a workspace that allowed you more private content and files. I won't dwell on that, but we can answer more questions if needed. There are otherwise 3 kind of views that you will see when you're working in protocols.io, and I'll quickly scooch through these. So file manager does exactly what it sounds like and is the location where you can see the files in your workspaces. You can drag and drop content in there, you can arrange things in subfolders, etc., etc. Workspaces are kind of a profile view of a group area where you can invite colleagues, collaborators, either co-located with you or from anywhere else geographically, to work in that space together with you to collaborate on methods. And then the editor is the place where you create the protocols themselves. So here's just what the file manager looks like. It's nothing particularly scary. It's exactly what it sounds like. You can have subfolders over on the left. So here, this one's just got example group, but you could have other folders within this folder. And there's a protocol and a document, and I can drag and drop in docx or PDF or any other file type that I want. When I click on one, I can see a bunch of options on the right without opening the file. And you can move things around, dragging them around. If you have more than one workspace, which I have quite a few, you'll see those down the left-hand side. So it does support absolutely any file type. You can drop some content in here. You can also archive and export content connecting to lots of different other external locations of your choosing. And it's all very secure and highly backed up. So you don't need to worry about the content that's on there. There we go. And this is just to show when it's a kind of a larger space, what the files will all look like in there. You can see when they've been versioned, the version numbers and the dates and the different owners. So this is a large group that we set up for the coronavirus method development community. So that's this workspace on the left. And if we click on that, oops, sorry, it's jumping around. You'll see that's how you can toggle between the file manager and go to the profile, which is more kind of the workspace view. And this is then what the workspace view looks like. And within the workspace, you can access the publications, you can write up and about to kind of explain what that whole group community is, see how many members, start discussions with people, either a high-level discussion or about a specific protocol if you trigger a discussion on a protocol, add links to resources and news and so on. So it's a really good community place where you can actually collaborate on content around a specific theme. So this could be a high-level theme, or it could be just for your research group and so on. And the workspaces have administrators, and there are a lot of different settings. So you have a lot of control whether your workspace is public and anyone can join, or if it's private and internal. So you can select exactly who is invited and allowed to join your workspace. And you can make that lots of different ways, public, kind of open to anyone, or by request, or actually just by invitation and send people a link, or invite them by their email. And then there are tons of other things that you can configure within there. But ultimately, it's a place to have the content that's shared between a group of people very securely. It also has a task manager and reagent manager, so you can set up things for that group to all have access to the same information and use that for the protocols. So mentioned you can export, so that when you're in a file, you can export all the content to lots of different external places. So obviously, although we have lots of archives and mirrors of content going on, if you want to make sure that you're also syncing your own content this way, you can. We also have this now. I don't think I've updated this screenshot, but I need to, because you can export directly to OSF as well now, which is not shown here, but it's something that you can do. Making a new protocol is incredibly simple. So when you're on that that file manager view, you'll click the plus at the top and you can pick protocol or collection or document, whichever you like. If you click protocol, then a new protocol in the editor will jump up. You will give it its title and you can just go ahead and start creating a protocol. So this is then how that editor view looks when you're editing a protocol. You can just add steps very easily, and sections. So we've got a first section here, which is color-coded purple, and then a second section, which is color-coded blue. So it's all in your control to set this up however you like. You can add components, which are the variables. So we can have a volume component, 200 milliliter, and we select these from on the right-hand side here. Volume, time, temperature, etc., all different variables of things that you might want to set. If you use the volume one, there's an option to scale later on. So if you have a master mix, you can scale up to generate something at higher volume and all of those kinds of things. You can put these critical steps on the left-hand side to show you at a glance what's coming up next. If you've got a pause point, and add in reagents, all down to the kind of catalog number level. All of these things will help with the reproducibility of the protocols that you're creating. Concurrent editing, very much like a Google Doc. In this one, there's only my little face here, so there's only me in it. But if I'd shared this with colleagues and they were editing at the same time, you would see them there and you could see the edits. You can check the history to see what edits anyone's made. Description, it's important to add an abstract. This helps with findability of the content. You can include and insert videos or lots of images. So this is content that you would be hard pushed to put into a research article to help with actually performing an experiment can go into protocols.io really easily. So you'll see the various options on the toolbar of the editor. It's very, very easy to either put the video in directly or embed code. If you have it somewhere like HTML, like YouTube, you can embed the code that will pull in the viewer from YouTube. Once you've made your protocol and you want to share it, you can either click share to very selectively add people or emails or a workspace, or just generate a private link and select who can see that file. You can also presently have get DOI or publish, which are different ways of obtaining a DOI. We will be moving towards just having this publish option with a DOI as the main way forward. And that means then you can put the DOI into the research articles as you're creating them and submitting them for review, and it will all be in there as the paper goes through the process. But it's also useful just to share directly with the DOI. It's a permanent link. Once it's published, you can make new versions, but you won't be able to change that version. And once they're on, it's very dynamic. So you can add, any viewer can add comments to a protocol at any given step or at the level of the whole protocol. And the author, the owner of the protocol, will see any comments. So in this case, if it's while it's still being created, you can edit according to the comments and address any errors. But if it's after publication, people can ask you questions about how to perform that experiment, and the authors will come back and answer them. And if the comment is made publicly, then that becomes a live FAQ, if you will, so that anyone else will benefit from that back and forth discussion about how to perform the experiment. It's really pretty cool because by pulling the method out from the research article, you really can facilitate cross-disciplinary collections. So this is a lovely example. The top left tweet is a researcher from Chile who was looking for an experiment using RNA for RNA extraction on primary cortical neuron cultures, so brain cells. Then Elena, a student at UCSF, I think, came along and suggested a protocol that was up on protocols.io. And when we looked at that, it turns out it's from a GigaScience paper looking at 3-spine stickleback parasite, so a fish parasite. It seems almost impossible that those researchers working on brain cells would have found these researchers working on fish parasites as a location to find the research approach, the experimental approach to take for their RNA extraction. But because the protocol has been separated from the research article, there's been this amazing kind of connection between research disciplines. So it can increase discoverability of the research that you're doing, certainly increases the reproducibility, facilitates connections, enables the reuse, and ultimately just enhances the value of the research that's being performed. I'm not sure we'll have anyone here who would be likely to use protocols.io for teaching, but it can be used that way. And we certainly have some great examples of labs using protocols.io, providing instructions for protocols that they want people to perform. But similarly for computational classes, I've not shown any examples, but you can very easily put in code and computational pipelines and workflows that way into protocols.io too. Pretty much almost the last slide, we do have an import service. So this is a way, if you're finding getting started with protocols.io to be having a very high activation energy, say this can be a way to ease in. If you have an existing protocol in PDF format or in a Word document, you can come to this web page, we enter protocols, the URLs at the bottom. We'll share the slides later. You can come here and upload the file. There is a fee for this, but If you are at an institution that has a premium license, you will be able to access a code that would waive the fee. And they're not very expensive, to be honest, for getting the protocol in. So we then have editors who are trained scientists who would upload a version of that protocol for you. You'll have a chance to review that. And then ultimately, once we have it to the level that you're happy with, we would reassign it to you privately. So then you could publish it or share it with colleagues, or add extra things like video files and so on to enhance it. And it can just be a good way of seeing how we can put your content in for you, how it could look presented on protocols.io. So please check that out if you think that might help you get started. And that is the end of the slides. We only have a few minutes for questions, but we'll probably stay on a few extra minutes. But if you did want a more detailed demo, there is a page, protocols.io/help/demo, where you can sign up for a one-to-one demo or a demo to your group, or feel free to drop us an email if you would like more of an overview of functionality at some point in time, and I'm sure we'd be able to set that up for you. But I'm very happy to take questions at the moment if anybody has any. Either pop them in the chat or into the Q&A. I can see Lenny's popped a few of the links into the chat already as well, so you can have the slides, and all of these links are in there too. All right, Lenny's going to just give me the tricky questions. Often we are asked what happens if you go bankrupt, which is a very good question. We hope that will not happen, but we have taken that concern very seriously and implemented a lot of checks and measures. So any public content is archived and mirrored in multiple locations. But even better than that now, we recently received grant funding from CZI, the Chan-Zuckerberg Initiative, and we now have with them a code escrow agreement. So should anything ever happen to our code, they would be able to open up that open source version of the protocols.io code. And we'll be working with them as well to better capture, archive and mirror the dynamic content. So the video files, the comments, all of the kind of add-ons that we have on protocols.io that grow with time. And versioning, etc., so that all of the content will be still available that way. But all the private content is also mirrored and captured on other locations. So users should never lose any of their content, even in the worst eventual potential scenario. Lenny, feel free to add more if I missed anything. No, that's helpful. That's there's our FAQ on that question. We will need to update that one soon then. But yeah, please check out the webinars page for more detailed webinars that will delve more deeply into functionality if you're interested in that, or feel free to sign up for a demo for that more kind of hands-on how to do something if you bring a scenario. We also run clinics, so if you're kind of struggling with a particular functionality and you want to join one of the clinics, you can just jump on that Zoom and ask any questions then. But I think we're at the half hour. If no one has any questions, we'll probably end there. Maybe, then if you want to turn off the recording, Lenny.